Hello and welcome to another pod. Today we're going to look at metabolism and energy production. Now this is still part of the anatomy physiology side of the house, but this time it's focusing on any cell with a mitochondria has some part to play in energy production. The lessons that you'll hear will be broken up into two parts, and we're going to go through what metabolism is and how those reactions of metabolism end up being utilized for energy. Part 1 will be an introduction to the concepts and then focus purely on the carbohydrate side of the house. Part 2, what we'll find out then. So when we look at the term metabolism, what it means is a series of chemical reactions that occur within the body cells, or occur within the body itself. And the end goal is to create substances from the foods that we eat that can be utilized to generate energy. That's the whole idea. So the generation of energy can be done in the presence of or the absence of oxygen. Now each one of those has slightly different processes and slightly different byproducts that are produced that we'll go through in detail. But just so you know that it can be produced differently in the absence or the presence of oxygen. What else metabolism does when it breaks down the foods that we eat, carbohydrates, proteins and fats, is it wants to make substances that can be utilized in the production of our energy. That's the whole idea. So when we look at the pathway of metabolism, there's 3 steps that we'll cover off on later, but it doesn't matter which compound we end up using for starters, all the different nutrients will get broken up until eventually they can converge onto the same pathway itself. So they'll end up at the Krebs cycle. So they all have a different start, but then from the Krebs cycle, they'll go through the same process in the production of ATP, or our energy itself. And that is obviously in the assumption that we're utilizing oxygen, which we'll explain more. Now, different types of reactions that can occur in order to assist us in this production of energy. First reaction is we have an anabolic reaction, it's like a building process. This is what happens when we store things. So the simple thing is we take glucose, or a similar molecule of glucose that's small, and as we turn it into glycogen. And glycogen will be stored itself. So we talked about this process a little bit when we looked at the endocrine system, but that reaction is called an anabolic reaction. The other type is a catabolic reaction. So when you talk about something catabolizing or a catabolic reaction, it means that you're taking a larger structure and breaking it down into its smaller components. And when you break it down, your end goal is to utilize that small compound in the process of producing energy. And as you'll find out, energy is 2 different forms. You've got ATP, which can be utilized by the cells, and heat. Just 2 different reactions that happen in the body in order to assist this whole process. And we'll refer to them more as we continue. So the 3 steps that we talked about for fuel metabolism. It starts right at the start with a disassembly of micronutrients. Okay, so it will get the nutrient compound and it will break it up. It will break up that compound, and then if it needs to, it will build different compounds. And you'll find out all about the different content that it builds. In the end, these different compounds that are created from the breakdown of our food will be used in the formation of adenosine triphosphate. So that's the whole process as it goes through. So we have a massive advantage that a lot of the things that we eat are made of the same substances. We've got carbon, hydrogen, and we've got oxygen. So they're all very, very similar substances that we consume. From there, we have a whole bunch of coenzymes within our body. So this entire process is assisted in the presence of coenzymes. Now, obviously, we know an enzyme is something that creates a catalyst or forces something to occur. So when we have all these broken down nutrients, it is the coenzymes and different steps within specialized organelles of the cell that allow those micronutrients to be built into special compounds, and those special compounds be utilized to transfer into the end product, which is adenosine triphosphate. That's what the whole purpose of turning what you eat into energy. Now, obviously, if you've listened to my podcast before, we're going to go into a lot more detail than this. This is the overview, so you understand the steps. Now, adenosine triphosphate is the high energy molecule that all of our cells use. Okay, it's the main component of energy that is utilized within the cell. It is a compound of adenosine, ribose, and then it has 3 phosphate molecules. Now, it is the breaking of the third phosphate molecule on the end of the ATP that generates energy that can be utilized by the cell. So as you'll learn later on, we have 2 substances within our body. We have adenosine diphosphate, and then we have adenosine triphosphate. Adenosine diphosphate, 2 phosphate molecules, can't be utilized to produce energy, and it's in its innate form until it can pick up another phosphate molecule, free-flowing phosphate molecule, and then it becomes adenosine triphosphate. And from there, it can be utilized as energy for the cell. So speaking of the cell itself, when we look at the cell, if you remember right back for when you learnt about the cell, most of our cells in our body have a whole bunch of organelles within it. And the ones that don't, we're not referring to in any of these processes. So when we look at the cell structures or the organelles in relation to energy production, there's 2 areas of the cell that we're interested in. We're interested in the cytoplasm or the cytosol. Now that's the area between all the organelles within the cell, so the fluid aspect of the cell itself. Okay, so that's the first area that we're interested in. The second area is the mitochondria. Now mitochondria, as far as organelles go, are extremely fascinating. They have their own special DNA, but they're also the absolute powerhouse of your cell. Looking at the mitochondria, you'll see that it's broken up into 2 distinct membranes. So we have an outer membrane, which is this capsule, and then we have an inner membrane. Now the inner membrane, when there's only one like there is in this picture, is referred to as a cristae. Now the space within the inner membrane is what's known as the matrix, and the space between the inner membrane and the outer membrane is just called the inner membrane space, or you could call it the outer membrane space. Okay, depending on where you read, it will say different things, but we'll know that it's on the outside of the cristae, on the inside of the outer membrane. 2 distinct structures that play a unique role in the overall production of ATP. So let's move into the carbohydrate chain of metabolism within a cell until we get up to the actual citric acid cycle. So from here we're going to follow a carbohydrate that we eat, and we're going to follow that process right from the start until we get to our end production of ATP through its final stage all the way down to the production of bulk ATP. From there we'll finish part 1, and then we'll move on to different aspects of metabolism within part 2. So starting off this cycle, we'll look at the carbohydrate itself. So when you have a carbohydrate and you eat it initially, so you think of anything that's high in carbohydrates, what happens is you end up with a large molecule. Common one that we have, not the only one, but it's a polysaccharide. So what that means is it's a number of different glucose molecules all put together. Now as soon as we put that into our mouth, our saliva produces allimase. Now allimase is a special enzyme that is used to break down large carbohydrate molecules into smaller ones. So then we get our dextrin, our maltodextrin, and our glucose. From there, the saliva does its job. We've done our mechanical digestion. We've moved down into the stomach. Obviously, that's a very acidic environment, but more importantly, that just sort of helps break down and loosen up the molecules that we have. Okay? That's it. From there, the molecules will move into the small intestine, and then we have a new series of different enzymes that are produced within the pancreas or free-flowing within the small intestine itself. So again, we have allimase, different sort of allimase this time from the salivary, but still the same process. It will turn any leftover dextrin into your maltodextrin or your lactose. Okay, so they're basically slightly different, but it will break it down into smaller sizes. From there, maltase, lactase, sucrase, different sorts of enzymes, turn those slightly larger molecules into glucose molecules. Okay, glucose, fructose molecules. From there, the glucose molecule is small enough to get into the bloodstream. From that point, glucose will go to the liver, and the liver will start its process of turning that glucose into a usable form, either storing it for later, changing it into glycogen, whatever it is. That's where it's all done within the liver. Okay, so that's the carbohydrate side of the house. Once we have a simple glucose molecule that can be utilized by our cells in order to produce energy, once it's in the bloodstream, it's free to be picked up by our cells, obviously in the assistance of our insulin. Once it gets into the cell, it goes through a process known as glycolysis. Now, as you know, a lot of words obviously have similar meanings. So glycolysis is simply the breakdown of glucose. Now, glucose itself is what the body prefers as its energy source. So when the body can use glucose, it will. Now, there's certain parts of the body that really rely on glucose itself, like the brain, and only in times of starvation will the brain use anything else but glucose as energy. But during glycolysis, glucose is turned into those special compounds that we spoke about that will aid in the further steps. Okay? Now, the interesting thing to note about glycolysis is it is an anaerobic process. Now, that doesn't mean that it's what used to be known as anaerobic glycolysis, because there is no aerobic glycolysis really. All it means is this particular step doesn't require oxygen, it actually produces oxygen. Okay? So, if we get our special glucose molecule, and that's represented by your C6H12O6, just for interest, what happens is it combines with two NAD. Okay, now we'll learn more about the NADs and the NADHs, okay, but in the presence with glucose and two NADs in the cytoplasm, it goes through a chemical reaction and it actually turns into two pyruvates itself. Okay, so two pyruvic acids are produced. On top of that, we also have two NADHs and four hydrogen protons produced as well. In the same process, that chemical reaction actually allows for ADP and two phosphates to become two ATPs of energy. So glycolysis is responsible for producing two ATPs and two NADHs within the cytoplasm of the cell. Now, the most important thing that is produced from this whole process is actually the pyruvic acid. Okay, so this isn't the only step that occurs in order to produce energy, otherwise it would be very, very ineffective, because so far we've only produced two ATP. And normal everyday processes can use thousands, if not millions, of ATP just to be conducted. Okay, so two within every cell is not really going to cut it. So let's find out what happens to those chemical substrates, pyruvate and 2-NADH at this stage. Let's find out what happens with them. The pyruvate can be broken down further to produce more energy. Now, it's this step where your anaerobic conditions and your aerobic conditions come into play. Now, things to note about the two different ones, things that most people probably wouldn't understand, is when you're in the anaerobic condition, in the absence of oxygen, you're actually able to produce more ATP faster. The only problem is you're limited to about 1.5 minutes, 2 minutes maybe, that you can continue to do anaerobic style of breakdown of pyruvic acid before the body will have to stop. If you use the aerobic side of the house, this is slow and methodical, but it produces a lot more ATP, and it also doesn't produce any byproducts that makes the body have to stop when using carbohydrates anyway, but still, it doesn't create any byproducts, which means it can happen all the time, all day, day and night, non-stop. But it's slower as far as yieldage. If you were to measure the amount of ATP produced in a minute under aerobic and anaerobic conditions, the anaerobic would produce more in the minute, but the aerobic would obviously produce more within the hour. That's essentially the way to think about it. So, looking at the aerobic condition, because that's the one that we will follow right through the next step. So in the presence of the aerobic condition, the pyruvic acid connects to coenzyme A and another NAD+. Now, when pyruvate is broken down, we lose a CO2. So that's part of the process where a CO2 comes from. But as it is broken down into a substance known as acetyl A, produces a CO2 and it produces an NADH coenzyme, if you will. Okay, so that's what happens. Now, it's the acetyl A that is the only substance that is able to enter the Krebs cycle. Okay, so that's why aerobic condition has a yield of more. So this process here is often contributed to just being part of the Krebs cycle. Okay, but the reason that I've broken it up is because if it's not done aerobically, it will never get to the Krebs cycle. So therefore, it's a different substrate step that is the changing of pyruvic acid into acetyl coenzyme A. And as a byproduct, we produce CO2 and we produce one of those other special compounds that we spoke about, which is NADH. Okay, so that's the aerobic side of the house. If we were to swap that condition, and now we're to put the same person producing energy into the absence of oxygen. This happens when you have to exercise a lot, but you do not have time to use the oxygen. Now in the next lesson we'll talk about the muscles and how they help buy you time in this process. But what happens anaerobically is your body produces lactate. And in that production of lactate, it doesn't actually produce any energy. But when that lactate is transferred back to the liver, it goes through a process of becoming pyruvate, becoming back to glucose, reverses the process, allowing more glucose to go through that initial glycolysis. So this is known as the Cori cycle. And this is an easy, very simple representation of what happens in your body when you're using anaerobic conditions in order to produce energy. So from the liver, we know it produces glucose. Glucose goes to the muscle. Muscle, as we just figured out just before in glycolysis, produces pyruvate. No problems at all. In that process, you produce ATP, 2 of them. That pyruvate, because there's no oxygen, turns to lactate. Lactate goes back to the liver, again, back to glucose, it goes back to the muscle, and this cycle continues. Heaps of lactate molecules are being pushed back to the liver, and then turned into glucose and pumped back out to the muscles in order to go through glycolysis, yielding those 2 ATP each time. So that cycle is rapid. Lots of lactate is going around the body, getting broken down back into glucose. The body's going through glycolysis back to back, producing those 2. But it has to stop eventually because the lactate's going to build up and it makes the muscles sluggish. Okay, so it builds up, that lactate builds up the acidosis and it builds up the sluggish muscle, and the muscle can't continue contracting in the presence of lactate. It needs to stop in order for oxygen to come in, help break down that lactate further, pick up those hydrogen ions and settle down the whole system before you can go at it again. So that is the anaerobic side of the house. That is why you yield more ATP within a minute, but you can only do it for a little while. Okay. I think it's time that we jump into what's known as the Krebs cycle. Now, this picture here has a little bit of the Krebs cycle in it, but this is your entire body's reaction and all the coenzymes that are involved in all the production of essentially a lot of different things. Okay, I only put it up there so then you know that a lot of people will complain about the tricyclic acid cycle as being complex, but that cycle is only a very, very small part in the overall picture of metabolism and energy. So when I explain it and you think it's getting a little bit too complicated, just remember this picture and realize that I have broken it down further into digestible conditions, okay, making it a little bit easier to follow for you. Okay, so don't shoot the messenger. Right, let's have a look at this citrate cycle. Here it is. So you may be thinking, well, this one's just as confusing, but don't worry, I'm going to break it down for you step by step, and we're just going to focus on the important parts of the actual Krebs cycle. And when I say important parts, I mean the ones that produce outcomes that can move on to the next step. That's essentially all we're going to look at within the Krebs cycle itself. So as already been mentioned, this represents the aerobic side of the house. So up in number 1, we see that pyruvate being converted into acetyl coenzyme A, and at the same time producing that NADH and hydrogen proton. Now within that same step, we've also produced an ATP. So for each pyruvate that's produced in that little precursor step, we've produced an ATP. Now from one glucose molecule, we produce 2 pyruvic acids, which means for every time we talk about how much energy a glucose molecule produces, think about this whole thing being doubled. It's the best way to think of it. Okay, so an easy way to look at this chart is to focus on the carbon molecules themselves. So glucose, as I mentioned at the start, starts with 6 carbon molecules, and then it becomes pyruvate, which obviously has 3. When it's oxidized and becomes acetyl coenzyme A, it loses one of its carbons, and that gets released as CO2. So now we have a 2-carbon acetyl-CoA. Too easy? The Krebs cycle, unlike the glycolysis, occurs within the matrix of the mitochondria. So this will actually go through the citrate membrane into the matrix, and that is where the Krebs cycle is conducted, within the matrix of the mitochondria, just as a point to note. Either way, let's start and follow it around. So acetyl-CoA enters the Krebs cycle, and then it is combined with citrate. Your citrate acid is the same acid that you'll find within your lemonade, just out of curiosity, so it's no different. From there, it goes through its citrate cycle. That's why it's called the citrate cycle. So it will follow itself around. By the time you get to number 3, you'll come up with a breakdown of different coenzymes breaking down the citrate and releasing more of these NADH and hydrogen protons, and at the same time releasing CO2. So you're losing 2 carbon molecules, and you're producing, transferring that NAD plus into NADH and hydrogen proton, and that happens twice. Okay? At the same time, we produce a little bit of ATP, right, in the presence of GTP, but essentially it's the same thing. Okay, it's energy that can be used by a cell. So we produce a little bit of that, and then it continues around its cycle. You get to number 4. By this stage, we've produced an FADH2 and another NADH, hydrogen plus. Now, there is a reason why I'm referring to these particular structures, because it's these structures, the NADH, the FADH squared, that will travel to the next stage of your energy production. Okay, your electron transport chain is the next stage, and that is what these particular chemicals will travel to in order to produce most of your ATP. So let's just recap our journey so far from that glucose molecule. Okay, so that glucose molecule has come in and it's gone through glycolysis. The glycolysis process has produced 2 pyruvate acids, 2 ATP, and 2 hydrogen ions with electrons. Now they're in the presence of your NADH and your H2. Okay, so that's what is produced. Then the pyruvic acids have gone into your citric acid cycle, and the outcome of that is the production of 10 hydrogen ions and their electron, and that's in the presence of NADH, hydrogen protons, and FADH2. They're the free-flowing hydrogens that we're going to utilise. And it's also produced energy in the form of GTP, okay, which is about the same as saying the 2 pyruvic acids have to produce 2 ATPs of energy. So at this point in time, after all of these steps, now glycolysis technically has 10 steps within it, okay, I just broke it down for you. And then we have an additional 8 different steps within the citrate acid cycle. So all these steps has essentially produced 4 ATP molecules, but more importantly, it has produced 12 of those special hydrogen atoms with their free electrons. Okay, that is the important part of this whole cycle, because it's those hydrogen atoms and NADH, FADH that is actually going to travel to the next space and yield most of the ATP that we find within our body. So let's get there. Let's talk a bit about it. The electron transport chain is the last stage of metabolism. So as mentioned, you only get to this stage if you can go through the Krebs cycle and produce all those NADHs. The initial ones can transfer over directly, but a lot of the times they're needed within the Krebs cycle itself. So it pretty much goes through the process as we described it. Okay. Now it's the electron transport chain that produces most of your ATP. Huge amount, 90% of the ATP is generated in this one step. Now this step takes place in utilizing that citrate membrane of the mitochondria. So at the moment, all these chemicals are on the inside, in the matrix, and they go to the membrane in order to react this process. Now the process looks a little bit like this. Now as a little bit of a disclaimer, a lot of this stuff that we mention in metabolism and energy production is absolutely known. Okay, so glycolysis, known. The difference between aerobic and anaerobic, that's known. Okay, been proven lots of times. Most of the process in that citric acid cycle has also been proven and is known. Now there's still aspects within it that are a little bit still being tested under investigation, if you will. Okay, now this electron transport chain, this essentially is up for massive debate and is still being researched at this stage. Okay, still being researched at this stage. So what you're about to hear about the electron transport chain is like the cutting edge of now, right now. This is the current theory, but it's still within the scientific community under investigation. Now, unlike this particular thing being under investigation, the other thing that's a hot topic that people love to argue about is the amount of ATP that is actually produced in this entire process. Okay, you can hear within textbooks anywhere between 28 all the way up to 38. Now the mean, how much you hear about, is the 32. So far we've got 4, which means this part here turns your leftover NADH and your FADH squared into 28 ATPs. Now that is based on the assumption that your cell and your mitochondria is optimal condition, and the glucose that you ate is optimal, and everything else works perfectly. Okay, we all know the human body is very different to that, and there's a lot of competing things for, especially hydrogen protons, especially electrons, which we'll speak about. So this is not an absolute, but it doesn't really matter, because this whole thing that we're talking about is an interest based. It's really good to know, but I don't think it's going to change your practice that much. Okay, but we'll get right to it. So on the inside of the cell, within the matrix, we now have a whole bunch of hydrogen-based enzymes and their electrons as well. And now these particular ones are transferred from the tricyclic acid phase to the membrane itself, and they're transferred by, again, coenzymes. Everything to do with metabolism and energy production is produced by coenzymes. Now, things that you need to know about hydrogen itself. When we talk about an hydrogen atom, it has a hydrogen proton, just the H+. Every time you see H+ written anywhere, it refers to as a hydrogen proton. At this stage, we have a free-flowing electron as well. So what happens a lot of the time is you'll have like 2 free-flowing electrons, 2 hydrogen protons, and an oxygen molecule, which is represented by, that half O2. Okay, if you're ever dealing with it in the blood and it says O2, it's actually 2 oxygen atoms connected together. So a lot of this hydrogen protons will combine, and you'll get a byproduct of water within this stage. Normally speaking, if you're just dealing with diffusion and you're just dealing with normal gradient curves and things like that, for starters we had a high concentration of hydrogen in the inner cell because of the presence of the NADH and the FADH2s. So as the breaking of the electrons, the electrons, as passing from the coenzyme to coenzyme, essentially opened the voltage gates of these ion channels, hydrogen ion channels, which forced the hydrogen out. No problems at all. It was easily done. You had a concentration gradient and a actual electrical gradient as well within the membrane potential. So hydrogen easily flew out. But now, as a result, the electrons are essentially done their job. They've opened up those gates. The hydrogen's flown out, and now you have an extremely high concentration of hydrogen protons on the outside of the cell and a very low on the inside of the cell. So you have a concentration gradient, and you also have an electrical gradient. So this area here is bound by the laws of diffusion, okay, and the laws of drift as well. Okay, so you've got that. You've also got a slightly acidic environment on the outside of the cell as well, because remember, pH is represented by these free-flowing hydrogen protons. So that's the current state of the outside of the membrane, so outside of the citrate itself. So we're not outside the mitochondria, we're just on the other side of the membrane itself. Okay. Now, this whole presence of hydrogen protons are obviously sitting there, but the voltage gates, the purple ones, aren't open. Okay, they're just not open at all. They're not open for business because they're not being charged. They're not being opened by coenzyme oxidation and the release of those electrons down the chain. Okay, so they're essentially stuck. And they're stuck until the concentration can be drawn to a very, very specialized protein structure called ATP synthesis. And once the hydrogens congregate, if you will, get drawn to the ATP synthesis, the hydrogen ions are forced through that actual channel, ion channel, and it's that forcing through that actually produces ATP. Now, here's how it works, which is pretty unique and very, very cool in structure. So I want you to think of ATP synthase, the actual protein, as a turbine, okay? And the hydrogen protons as water moving a windmill or wind moving a turbine, whatever it is. As they pass through the ATP synthase, they cause the base of the synthase to rotate and rotate and rotate and actually generate movement within that particular area of the protein itself. Now, within the capsule at the base of the ATP synthase, you will find a whole bunch of ADP and free-flowing phosphate ions. And it's the spinning of this turbine that generates movement of these molecules that causes it to stick together and create ATP. Okay, so what they say comes out from one glucose molecule all the way through those 10 NADH, produce 28 ATPs at the end of the electron transport phase. And all this is happening right at the membrane of the matrix within the mitochondria of the cells. So every cell. The picture in front of you represents a very small percentage of the overall membrane that contains these structures. So each time it comes through, it is producing all of this. And then NAD and FAD are back within the system in order to go back to the Krebs cycle to pick up more hydrogen as well within the actual citrate itself. And the process will continue and continue. So that is the production of ATP in a nutshell. Remember, this one was focusing on carbohydrates itself. So hopefully you got something out of it. Like I said, this one here is purely interest based. It's not going to revolutionize any of your practice or anything like that. The second part of the series, we're going to find out what happens to fats, what's proteins, and then we're going to look at the overall metabolic rate and things like that of the human body. Okay, so until next time, take care.